Thanks, yeah. So this afternoon I'd like to take this opportunity to talk to you quite loudly, it seems, about my work in developing sustainable algal biofuels. And these are experimental investigations undertaken in the fuels and combustion laboratories of UCL Mechanical Engineering in very close collaboration with the UCL Institute of Structural and Molecular Biology and also UCL Biochemical Engineering. Okay, I'm sorry, the format of the slide seems to have been a little bit squashed. But it'll be okay. Anyway, so I want to start out by talking a little bit about future fuels and our whole approach to developing them in collaboration with other researchers. And then I want to look a little bit more at the advantages and disadvantages of using algae as a feedstock for developing these future biofuels. And then I'll talk in a lot more detail about the two approaches that we investigated experimentally to try and overcome some of these disadvantages associated with algae as a feedstock for biofuels. Okay, so we're in a period of transition where we're going away from the liquid fossil fuels that we've been using for the past 100 years towards a whole new generation of different sustainable alternative fuels. And I think that when we're faced with trying to replace a resource that arose from a feedstock and was created according to processes that lasted millions of years, for us to try and replicate exactly the end product it's not realistic and it's not particularly desirable either. Instead, what I think we have, and for me as a combustion engineer, it's a very exciting opportunity to instead work with biologists, work with chemical engineers, work with process engineers to look at how we can best take a sustainable resource, so a photosynthetic microorganism, some kind of waste stream, and we can convert it with the minimum of energy inputs into a useful fuel that's been designed to be fit for purpose in terms of the way it releases energy in combustion systems, and also to minimize the amount of unwanted pollutants that we get out when we combust these fuels. So things like certain particulates and also nitrogen oxides, regulated pollutants. So considering algae in particular, so algae is it's a single-celled microorganism that lives sort of quite on its own, in a way, in large bodies of water, and it accumulates within the cell oils that are very similar to those that we extract from crops such as sunflower, rapeseed, and palm in order to produce biodiesel. That's those oils reacted with various alcohols to get a fuel that's useful in modern diesel engines. And so one of the big advantages of algae relative to sunflower, etc., is that we don't yet eat algae. And so we're not competing with food resources. We don't need to use high-grade agricultural land to grow our algae. We can do it anywhere where we have some water. And that's another advantage of algae. We can actually incorporate into the growth of algae remediation of various waste streams. A little bit of work that I did on the side, which I don't have time to talk about today, is looking at growing algae on various effluents from sort of power stations and things, using the CO2 generated in producing energy and putting that back into algae and growing a whole new sort of generation of biofuels. And you can also take wastewaters, which are high in the nutrients, and normally you need to treat those to get back to a level of purity that you can release back into the environment. You can use algae as a means of extracting those nutrients and then growing the algae itself. But there are disadvantages with algae. As I mentioned, it grows at very low densities in water. So where we have these oils, they're useful, they have an energy content, but they're trapped within the cells of the microorganisms. To get to them, we have to dry the cells, and that requires quite a large energy input. This harvesting of the oils and getting at what's useful is very energy intensive. And furthermore, once we get those oils out, they're not yet ready to be put into an engine or something. We need to treat them in the same way as we do with sunflower oils, etc. There are a whole range of sort of chemical processes that they need to be subject to before we get our final product. And so that's two quite energy-intensive steps that need to be undertaken before we have a final fuel. So that's where we wanted to come in and try and find alternative ways of using algae to avoid that energy-intensive harvesting step. So the first thing we did was look at algal slurry diesel emulsions. So what these are is basically standard fossil diesel mixed with some algae, which is still wet. It's still in, say, it's still sort of 80% water, only 20% dry algal biomass. And what we did is we took these emulsions and we burnt them in a modern diesel engine, very similar to what you might find in, say, a Ford Transit van or a Ford Mondeo. And we wanted to see if we could get sort of a calorific contribution from the dry algal biomass that was in these slurries, if in fact, despite the water being there and we haven't undertaken those additional processing steps, can we get the algae to burn and release energy? And so here are some very preliminary results of these experiments. So the plot on the left-hand side, this shows a heat release rate of some various fuels during a typical combustion process inside a diesel engine. So heat release rate really is just an indicator of how much energy is being released during the process of combustion at any given point during that combustion process. So the red trace that's 100% diesel. So that's burning normally, as you'd expect it to in a diesel engine. The blue trace is a control emulsion. It's just the water and the diesel, no algal biomass. And then in the middle, we have a green trace. That's the algal slurry and the water. So the algal slurry and the diesel. So that's the same amount of water, but with the added addition of the algal biomass. And what this tells us, the fact that we've got a higher peak there, that's indicative of there being a greater amount of energy release than in the control condition, where we just had the water and the diesel. So we're still analyzing these results, but that's a good sign. It seems to be that even though the algae has not been sort of reduced down to a dry state and we've extracted those oils, once it gets into the engine, once the combustion processes start with the diesel burning and everything, the temperatures are high enough to carry on and burn the algae and get some useful energy out of it anyway. Another thing we did to try and get a handle on whether or not these cells really were combusting as we wanted was take a look at the soot particles coming out of the engine exhaust. So the top image there, that's from just the control emulsion of water and diesel. And that's a very typical diesel soot particulate. You can see it's kind of an agglomerate of very, very small particles sort of kind of formed together in a sort of scaffolding shape. Now, the algae cells that we were injecting into the engine were sort of 2 to 3 microns in size each. So they would, if they were still present, they would be around about the same size as that particle, but with a very different morphology. They would look like a cell still. And so when we looked at the same particles in the case of the algae emulsion, what we saw is a very similar morphology to the control emulsion. There doesn't seem to be any evidence of those algae cells surviving and coming out whole in the exhaust. So hopefully that means they did burn and they did take part and release energy. So another approach we've taken is to look at how we can kind of design the fuel from scratch by using the ability to genetically modify microorganisms, photosynthetic microorganisms, to produce specific targeted fuel molecules. Okay, and we started in this particular project looking initially at one particular fuel molecule, geraniol there, over here, just above me. And so the biologists I'm working with, they were very keen that we look into this as a potential biofuel, because they found that this particular molecule, rather than accumulating within the cells of the algae, would be secreted into the media, the aqueous media in which the algae was growing, and could potentially then sort of be separated from that media without the need to dry the algae and to put into all that energy. You could have a system where you could just sort of scoop the useful oils off the top of the media. Now, a lot of the other work that I've done has been looking at how the molecular structure of a fuel impacts on the way in which it releases energy, and also the way in which it impacts on the levels of emissions that are formed. And so with this knowledge, I looked at that molecule, and I immediately, I could tell the biologists that we're working with, well, perhaps this isn't going to be a great fuel molecule. It has some of these features, these carbon-to-carbon double bonds, the branching, the alcohol group. These are things which aren't ideal in a diesel fuel molecule. But then I looked again at this map, and I could see that there's a whole sort of range of potential for subtly altering the molecular structure by introducing different enzyme pathways. And so what we did is we kind of put together this matrix of different fuel molecule structures where we did very sort of subtle, small changes to the molecular structure of these fuels. For example, changing the orientation of the double bonds from sister strands, reducing the number of double bonds entirely, or playing with the oxygenated functional group, adding more oxygen to the molecule, or just making the molecule larger, increasing the number of units that were kind of put together to create that fuel molecule. And we took these and we put them back into our diesel engine. And yeah, this is a bit of a mess, but we combusted them. And that gave us an idea of how they performed relative to each other, and also a reference fossil diesel. So the left-hand plot again is, this is ignition delay of the various compounds. So ignition delay is the duration between which you inject a fuel in a diesel engine and the time it takes before that fuel auto-ignites, before it starts combusting. And this is a very important parameter in dictating efficiency of energy release and also the level of pollutants that are emitted. Things like nitrogen oxides are highly temperature dependent. The higher the temperature in the combustion process, the more nitrogen oxides you get produced. And what we found is that making some very minor modifications to the structure of that initial fuel molecule, the geraniol on the far left-hand side, gave us a big decrease in the duration of ignition delay, bringing it down to be a lot closer to the reference diesel. For example, changing the alcohol functional group to that of an aldehyde functional group, down to geraniol down there, just changing that OH for a O, that brought us down to a fuel that was very similar to the reference diesel. We also looked at the emissions. On the right-hand side is the total mass of particulates produced by each fuel. And what we saw is that, again, very subtle changes to molecular structure could result in undesirable sort of consequences in terms of the emissions. So the squalene, which is a much longer molecule, that performed favorably in terms of ignition delay, but it produced a lot more particulates, kind of ruling itself out of the running in terms of how we progress this design of the fuel molecule. And so after these combustion assessments, we were able to go back and talk to the biologists we're collaborating with and say, well, we found two molecules, geraniol and farnesene, that they performed a lot better than that initial molecule you're interested in, the geraniol. And so can we take the features of that molecule which are contributing towards making a better fuel, can we take them and combine them and use this to guide the next stage of your genetic modification of these fuels? And yeah, there was a pathway, an additional enzyme pathway that could perhaps be added into these microorganisms to get to that more desirable looking fuel. And that's what we're doing next. We've got a whole sort of iterative process going on. And this is going to be the next step of combustion testing. I'll be testing this farnesyl, and we'll be moving on from there. So, to summarize, algae has a lot of potential as a source of biofuels because it doesn't compete with food resources. It already produces compounds which are similar to some of the things we already use as biofuels, but there are problems with it. You need a lot of energy to extract the useful materials, and you need to post-process those materials. So we attempted two sort of experimental routes to circumventing these problems, one of which the combustion of algae slurry diesel emulsions looks promising. We seem to be getting a contribution from the algae despite it still being there with all the water and everything that it grows in. And another exciting approach that we looked into was can we sort of design a bespoke fuel by working with biologists to exploit these genetic modification methods to target specific molecules. Thanks.
We certainly look forward to the real results and seeing this fuel then on the street in the cars eventually. Thank you. A question from the floor here is the first.
Very interesting research, thank you. What do you think of the shallow tank saltwater approach from the US Aquatic Species Program in the 90s? Is that still a viable direction for research, or is it dead for some subsequent reason?
I've got to be honest, I'm not entirely familiar with that. So that's the idea, is that growing algae in salt water, is it?
So you have very large shallow tanks,
Okay.
1 to 2 feet deep, and instead of going for the monocellular algae which are suspended in the water, they're looking for the stuff that looks like the hairy green crap that grows on rocks.
Oh, okay.
So the kind of slimy stuff, and you have an open tank ecology. So rather than going for the Monsanto approach where you genetically engineer the algae and then fill it with Roundup, it's multi-species, so as you get higher and higher up the food chain, you get higher and higher lipid content, and then you scrape the entire thing off with a machine that looks like a combine harvester, chuck it through a bioreactor, oils on one side, alcohols on the other.
Okay, that sounds really interesting. I mean, yeah, because it cuts down that energy requirement. for harvesting the algae when it's just floating in water. So that sounds like a very interesting, but I don't know if you'd lose that ability to design precisely the outputs if you've got kind of a whole ecosystem of different things.
Yeah, I mean,
Yeah.
it's a farming approach. The program ran until about '93. They were looking at producing a diesel for $25 a barrel equivalent.
Oh, okay.
And at the time, oil was $13 a barrel, so project was killed.
Yeah.
But it's kind of the path not taken on algae research, because they went to solve the problem of how you get the monocellular stuff out by just not growing monocellular stuff in the first place.
Yeah, absolutely.
So I was just wondering whether it turned out to be a dead end or whether it was a dropped thread.
It's
like
not something I've come across, so maybe it was sort of a dropped thread, and yeah, but maybe there's potential to sort of look at that kind of.
Fascinating. I will send you a couple of links.
Okay, that'd be great.
Later.
Thanks.
Thank you.
Thank you. More questions around? Yep.
As an alternative to the modification of for the fuel design, what importance is the structure of the surfactant in the algal diesel slurry experiment? So did you look at surfactant design?
Well, the importance of the surfactant was in getting sort of a surfactant package that would give us a stable emulsion for a decent amount of time, because obviously you can't be creating this emulsion and for it then immediately to sort of separate back into its layers, because you can't transport it anywhere. It can't be used in that respect. But certainly the surfactants themselves, they were sort of of specific molecular composition, and they did impact on the way that the whole emulsion combusted and I could see that in the results.
I see. So you could optimize based on different surfactant designs, even that type of combustion.
Oh, absolutely. Yeah, you could optimize the surfactant for keeping your emulsion stable, and also for the effects that it has on the combustion. Yeah, definitely. Yeah, there's potential to do that. At the moment, we were just looking at can we have a stable emulsion, and how we wanted to get the maximum amount of algae into the emulsion to try and get a sort of a positive result with as much confidence as possible. Thanks.
It sounds like a really fun research, and I was wondering, like, the fact that you are now kind of playing around with sort of developing the, let's say, perfect molecule. I was wondering, like, the sort of farming side of it, because something that sounds very interesting to me is whether the kind of molecule would be at some point quite easy to grow, so that, like, individual people or customer could actually have their own little growing bioreactor in their home or in their farm or in their building, whatever they are. So suddenly it becomes not just a kind of alternative fuel, but also completely opens up a different way of potentially accessing and fueling your car. So you're not like reliable necessarily to the kind of centralized distribution of fuels, etc. But you can basically do your own and then put it in your car. Is there like also that side of the research you may be able to do, like actually verify that maybe the final molecule is also simple and easy enough to grow? Or?
Oh yeah, definitely. I think because what we're trying to do with designing the fuel molecules is remove the need for these post-processing steps. So with the oils that they use at the moment, it's an industrial process again to react them with alcohols and produce biodiesel. But what we were trying to do is find a molecule that was being produced directly that was straight away suitable to go in an engine. So yeah, that would fit in really well with the model of people producing them.
Much easier to just go from the growing to
Absolutely, you wouldn't have to send it off to a refinery. You could, yeah, perhaps do it at home. Yeah.
So what sort of surface area do you think you would require in order to kind of replace a large proportion of the fossil fuels currently used in the world? I mean, on the one hand, surface areas on the planet itself could be desert areas, as you indicate. On the other hand, offshore areas. I mean, can you imagine offshore saltwater type of based algae production so that you don't compete with other land uses?
Oh, definitely, yeah. I think there's potential to do it offshore. I don't have sort of the figures and things on me, but I know the area. The area is huge. I'm probably wrong, but is it the size of Texas that the area needs to be, or is it the size of Europe? It's somewhere in between those two extremes. But it's huge. So it's not the solution to what we do to replace fossil fuels, but I think it's part of the package. And if we can be smart about the way we're using it, looking at these tools of genetic modification and things, or reducing the amount of energy that needs to go in to get a useful product, then it makes it more viable as an option, I think.